one of which is muscle glycogen. Now, one of the things that we recognize is that under static conditions, muscle glycogen only decreases fundamentally because of a lack of oxygen. But under dynamic conditions, even when oxygen is not limiting, as I'm about to demonstrate, muscle glycogen content will decrease. So here we've got a really nice data set published in 2013. And what you can see here is they've got exercise time to exhaustion, about over 180 minutes in some conditions. And these represent the percentages of VO2 max that the participants are working at. So we've got 31%, 64%, 83%. Now, what we don't know is where these sit, particularly particularly the 64%, in relation to what's called the lactate turn point. The lactate turn point being that transition from predominantly aerobic to predominantly anaerobic metabolism. We can be pretty confident that 31% is below the lactate turn point, and we can be pretty confident that 83% VO2 max is above the lactate turn point. This is perhaps the slightly more ambiguous one. But nonetheless, in all three conditions, they're below the VO2 max. So in all three conditions, they've not reached maximal cardiac output. They may have reached maximal stroke volume at about 83% VO2 max, but they certainly haven't reached a point where oxygen delivery is absolutely limited. They haven't reached a point where they are struggling to extract the oxygen at the cell. Yet what you see is even at 31% VO2 max, it's not marked, but at 31% VO2 max, you see a decrease in muscle glycogen content. You go up to 64%. So I would hazard a guess at 64%, they are probably still below the lactate turn point. You see a significant decrease in muscle glycogen. Oxygen isn't limiting. It's limited, but it is not limiting. And even at 83% VO2 max, so below the VO2 max, so certainly not solely relying on anaerobic substrate metabolism, certainly not in a condition where the cell is solely hypoxic, there's an enormous decrease in muscle glycogen content. And then if you tie that in with the other variable that we're looking at here is, look at the time to exhaustion. At 31% of VO2 max, the time to exhaustion is 180 minutes, and they probably stopped the test probably due to boredom, which is perhaps that more kind of perceived fatigability that Inoku was referring to earlier on, that kind of centrally orientated response. Then what we see at 64%, where they terminated at 120 minutes, and at 83%, where they terminated at 60 minutes. So these are all indications, particularly these two, are indications of a fatigue. There is a fatigue here because they terminated the test. I mean, we don't know whether that was the authors just terminating at 180 minutes or whether that was kind of an average of where everybody got to. But you can certainly get the sense of task failure at 64 and 83% VO2 max. But oxygen isn't the limiting factor, yet muscle glycogen concentration has decreased. And that's fundamentally, quite logical, isn't it? That particularly if I'm beyond the lactate turn point, I don't have that ability to resynthesize muscle glycogen. I'm going to be utilizing the glycogen. The only way I can start to resynthesize the glycogen is if I can go below the lactate turn point and probably below the lactate threshold, below LT1, and start to resynthesize glycogen, which is possible, maybe about 30%, very much depends on the individual, but certainly not for these two workloads here. So the net result is that despite the fact oxygen isn't actually limiting that much, muscle glycogen decreases because we've got an increased utilization of this highly abundant store. And if we go and look at this, which is from a paper in 2013, the authors make a really important point, which is this. Leading to an inability to translate the motor drive into an expected force, i.e. fatigue, develops. This is supported by observations of phosphocreatine decreases, along with an increase in free ADP and IMP, inositol monophosphate, following prolonged glycogen depleting exercise. So they've kind of gone to an extreme here, which is what happens if we deplete the muscle of glycogen? Well, not surprisingly, force decreases. We've already seen that in the previous slide. And if force decreases, that fits our definition of fatigue. But because that has decreased, the net result is, with a lack of glycogen, you now have to rely more on PCR for force generation. If you rely more on PCR, you're going to get an increase in free ADP. Remember that we said that the phosphates from PCR would be donated to ADP to give us ATP. But the ATP molecule is then acted upon by water, H2O, to generate, in the presence of ATPase, the enzyme, to generate energy. But that leaves us now with free ADP. And unless that ADP keeps being attached to phosphate, it remains free. And actually, free ADP is a really good indication of fatigue because that concentration increases. The same with IMP. Inositol monophosphate is a consequence of what is called the adenylate kinase reaction. And the adenylate kinase reaction is a reaction that allows the combining of ADP to ADP. So if we combine ADP to ADP, we've got 4 phosphates, so we get ATP plus AMP. The AMP is then acted upon by the enzyme adenylate kinase. That leaves us with IMP, and actually we end up with, by a slightly larger process, we end up with ammonia as well. So the IMP is also an indication of fatigue, because the only reason these start to increase is because we're using our substrate level metabolism at an accelerated rate. And in fact, the authors go on to state this: however, the energy deficiency theory in skeletal muscle fatigue is challenged by both in vitro and in vivo studies demonstrating a strong association between low glycogen and decreased muscle function, even after recovery periods where ATP levels would be normal. So in other words, just because you've got normal ATP concentrations, you've still got to have enough muscle glycogen available. Because glycogen plays an enormous role, don't forget, as does glucose, in things like neural function. So the idea of getting excitation, contraction, recoupling, the idea of the muscle to be able to generate force, yes, I can have ATP because that's fundamentally what I need, but unless I've got the glycogen and the glucose, actually the process by which those motor units are activated and action themselves starts to decrease. So glycogen actually, and the availability of glycogen becomes a really important factor in fatigue. Whereas actually, if you just looked at the ATP concentration, you may argue, well, actually, there isn't a fatigue there. Most of the previous slides that I've shown have shown ATP concentration remains relatively stable. But that's fundamentally because we are always trying to resynthesize ATP. That's the goal, really, in terms of metabolism. And we can see the glucose effect in this really nice figure that we've got here from this paper. So what you've got here is you've got repeated contractions at 100 hertz with and without glucose. And what they've done is they've infused it. So what they do is they fatigue the muscle here. So it's 240 seconds of and again, it's isolated muscle. 240 seconds at 100 hertz, that's 100 cycle, 100 contractions a second. And what you see is not surprisingly, the muscle becomes fatigued over time. You get quite a rapid decrease here from about 180 seconds to about 240 seconds, quite a significant decrease. Then over a 60-minute period, they reinfuse the muscle with a glucose concentration of 5 millimole, and then repeat the task. And what you can see is, pre to post, there is almost no change in that profile in terms of the amount of force that's being produced across the isolated muscle fiber. It's actually almost identical. Now look what happens though when glucose availability becomes limiting. So when glucose availability becomes limiting, this is pre, it's a similar profile. It won't be identical, but it's a similar profile. Okay, it'll be a different day, different condition, different muscle fiber they've probably worked upon. But look at the post. Look what happens when glycogen has not been given the opportunity to resynthesize. In other words, glycogen becomes limiting. So this is kind of an extreme way of representing this. Glycogen is limited, and in this case it's limited because we haven't got glucose. So remember, carbohydrates fundamentally is limited. Look at the impact. They were able to actually go in this example for nearly 400 seconds, slightly longer than they did here, but they couldn't even get to halfway, or the muscle fiber couldn't even get to halfway post the 60-minute period where glucose and therefore carbohydrate and therefore glycogen availability become limiting. So it's really indicative here because there is no indication that oxygen is limiting in this scenario. It is simply about the lack of glycogen. So it really lends itself to this notion that glycogen is a fatigue agent in the sense of, no, I'll rephrase that, the glycogen is a reflection of fatigue, and actually that we recognize that it responds to most clinical and sporting environments by showing a decrease because it's used in energy liberation. But the resynthesis of it, this is kind of important here. We have to provide the correct environment to resynthesize the glycogen. Now that's where things like oxygen does become important, because this is where the notion of, as we've covered in previous sessions, where we did a session which was on the notion of recovery and regeneration. If we think about kind of an active recovery, for example, an active recovery actually allows for the restoration in kind of more dynamic situations of muscle glycogen and actually allows us to kind of regulate the blood glucose concentration. Whereas if we don't have that kind of active condition, or we restrict glycogen or glucose availability, you can see quite clearly the consequent effect. And this example here is perhaps a little bit more difficult to see, but what we have in this schematic, we have kind of changes. These are kind of under the electron microscope that we can see here. So we've got different muscles, different muscle fiber types, but what we've got is A and C are pre-exercise and B and D are post-exercise. And what you can see is the kind of effect. It's not overly clear, it's never quite clear, but if you look at the availability around the fibers in terms of the storage of the glycogen, so the kind of the dark area represents our glycogen, significantly reduced post exercise, and you can kind of see it a little bit here. They've actually highlighted it for you on this slide here, so you can see the gastroc glycogen content, and this is the gastroc glycogen content, when post exercise. You can see that kind of significant change in its availability, its abundance. And this is quite nice to kind of reinforce this. Again, a relatively old study. This is Greg Gaetanis' group when he's working with Balsam and colleagues. So what they've got here is they've got repeated sprints. So they've got 15 repeated sprints, and they're 15 sprints, 6 seconds. And what they've done is they've taken a recording in the last 3 seconds, but they've done it in 2 conditions. They've done it in a condition where they've got low muscle glycogen and high muscle glycogen content. Now this is quite important, because these are only 6-second sprints, and they're 6-second sprints, so the predominant substrate under 6-second sprints in dynamic exercise, the predominant substrate is going to be PCR, no doubt about that. But what you start to see is when muscle glycogen content was low, look what starts to happen in terms of pedaling cadence. After about sprint 9, we get a rapid decrease in cadence, whereas actually when glycogen content was high, that doesn't really happen. So why is that the case? Well, remember that as we're doing these repeat sprints, and they've only given 30 seconds recovery, at the end of the sprint, you've got to restore the substrate that's been used, so it would be the PCR. So if we kind of work using Gaetanis' old work, which was they estimated back in the 90s that you get around about 50% restoration of PCR within 60 seconds. So what we're saying is, in 30 seconds, let's go roughly, you probably only get 25% restoration. So in 6 seconds, you won't have got full depletion. So 6 seconds, you probably, let's say, use 50%, you've only restored 25% of that 50% after the first sprint. And so you go into the next sprint, you're already starting to get a markedly reduced PCR concentration. So the net result is that irrespective of whether you've got high or low muscle glycogen, after a number of sprints, actually a very short number of sprints in this scenario, PCR is almost completely degraded, it's almost gone. So then there is that kind of increased reliance on muscle glycogen. And at that point, where glycogen becomes limiting, look at the force output. So this is a kind of a real eye-opener for us in terms of understanding that kind of connectivity between high-energy phosphates, which you think are the predominant substrate here, and actually having the muscle glycogen to support that. And this is a real indication of a fatigue-based response when glycogen becomes limiting.